Stanford University.
This is the final seminar of the quarter, so make sure to email me by next Tuesday about your course attendance, and I'll send out an email reminding you about that. So I'm pleased to introduce Adrien Treuille from CMU. He got his PhD working in graphics at University of Washington, working on some really cool crowd movement stuff, and is now a professor at Carnegie Mellon, has done some great work on crowdsourcing, and today he's going to be talking about next generation citizen science.
Cool. Thank you, Leslie, and thank you, Scott, for inviting me. I'm excited to talk here. And in particular, a lot of the research I'm going to talk about is actually joint work with Stanford. So I'm particularly happy to be talking here. So I'm going to talk about crowdsourcing science. And so what do I mean by crowdsourcing science? So we'll jump right in. The first thing I'm going to talk about is protein folding. So proteins, as you know, are one of really the key molecule to life. And they fold into these incredibly intricate and complicated shapes in our cells. And so formally, the protein folding problem is to take a sequence of amino acids, which is sort of the underlying description of a protein, and try to predict which shape that sequence of amino acids will fold into in vivo, in a cell. And so this is really considered one of the, understanding protein foldings is considered one of the fundamental grand challenges in biochemistry today, really in all of science. It's a key to understanding life. And this problem has required traditionally huge computational resources. So you know, folding at home came out of here at Stanford. People have been putting together large networks of computers to fold just a single protein. But the protein that you see here isn't being folded by an algorithm at all. It's actually being folded by an 11-year-old boy in upstate New York. And this is a game which we created called Foldit. This boy is actually one of over 300,000 Foldit players around the world who are competing and cooperating to fold proteins. So now I'm going to give you another example, RNA Nanoengineering. And if you think about this, this is actually the inverse problem of protein folding. So in this case, the shape is given, okay? And the key is to find a sequence, this time of nucleotides, which naturally will fold into this shape in the body. So it's the inverse of the protein folding problem. And this is also considered a very important problem for designing next-generation catalysts, drug-responsive control elements like riboswitches. And we created a game for this as well, which we call Eterna. So this is actually a molecular design interface, which we designed. And it looks a lot like Foldit, but it takes Foldit one step further, because every week, players vote on which designs they think are best. And then we actually synthesize here at Stanford top player designs and send back images of what the players designed to them every single week using high throughput biochemistry. So if you think about it, this cycle of sort of experiment and hypothesis result is really crowdsourcing the entire scientific method. So these are the two games I'm going to talk about. So Foldit was launched in 2008. It's a protein folding game. It has about 350,000 players, computational chemistry. And Eterna is a newer game. It was launched, just had its first year anniversary, actually, birthday. It's an RNA Nanoengineering game. It has about 35,000 players. And it deals with experimental chemistry in the sense that it's really an online design competition where we really build the RNAs. And so when we created these projects, we had this idea in our mind that, you know, basically it looked like this. So we were scientists, and we had this problem. And we were going to create a game to solve this problem and give it to players. And this seemed like a really cool and fun idea, and we didn't know what was going to happen. But if it worked, we wouldn't have to do work again for the rest of our lives. So it seemed awesome. But it turns out that that's not what happened at all. In fact, the moment we gave this game to the players, sort of injected it into reality, they started using it completely differently than we expected. And they were good at things that we didn't expect and bad at things that we didn't expect. And that changed the kind of science we were doing around these games, which then changed the kind of science the scientists were working on. So injecting the players into this game changed everything in a very nonlinear way. And all sorts of crazy little eddies came out of these projects. And so that's actually what I want to talk to you today. It's a very non-technical talk. It's basically 10 reasons to crowdsource science. And it's a bunch of stories about what happened to us when we created these games and handed it to hundreds of thousands of people. And there's few enough people in the room. I know we're being recorded, but I think it'd be OK if people just jumped in with questions. Yeah, I think that'll be more fun. So just jump in with questions, and I'll repeat them for the folks back home. OK, 10 reasons to crowdsource science. So the first reason is that games make us understand. So this is the Foldit interface, and this is the Eterna interface. And when we created these games, people ask us, like, why did they work so well? And I think that one of the reasons was because the computer scientists working on these projects had no idea about anything about biochemistry. And so in some sense, we were forced to do something very valuable, which is look at these very exotic and esoteric phenomena sort of like through the eyes of a child. And thankfully, we were working with fantastic biochemists here at Stanford and at University of Washington who had very deep and sort of clear understandings of how these objects fold. And so we translated those principles into a game. And the results are these interfaces that you see here, which look nothing like anything on the market. I mean, before we created Foldit and Eterna, this is what a real scientific interface looked like. Okay? And so our games are totally different. We actually tried to come up with sort of simple, intuitive, visual metaphors, borrowing a lot from the game community, which would express what kinds of manipulations are possible on a protein, what these forces feel like, and then also really just the repertoire of forces that determine how our proteins fold, and similarly for RNA. And so a very interesting thing happened when we created these interfaces, which is that real scientists loved them and were begging us to just give them the interface, forget the game. And so actually, there's now a top secret, it's not really a secret, expert version of Foldit, which has stripped away all the scores and the stars and stuff, which is being used across the country in biochemistry labs to do interactive protein folding, which really there was nothing you could do that on before. And similarly, Eterna, actually, we just got contacted by a pharmaceutical company, which said, we love this interface. When people buy RNAs on our website, we'd love for them to use this game so they can see what they're buying. And so I think this is very interesting, because what it means is that independently, these two games were developed completely independently of one another. Scientists wanted this kind of interface very much. And so I think that this is really possibly the start of a new field of interactive biology, which has a lot of implications, not just for games, but also for training and for education, and for how scientists working in these fields actually look at the phenomena themselves. So it's a very exciting thing that just came out of this research, really serendipitously.
Adrien,
Yeah.
What led you to think that you might be able to get a crowd to do this?
Well, we had, first, I guess there's a bunch of questions there, one of which is what led us to believe that any person could do this? And then another one would be what led us to believe that people would actually want to play this as a game? In the case of the former, we really had no idea. And I'm going to talk a little bit more about this. If you talk to biochemists, people getting their PhD in structural biology, they can often tell you this protein looks right or this protein looks wrong. That's a very interesting thing. So it seems as though with enough training, you can gain this intuition. And that was sort of the seed of the idea. Whether or not it would work as a game we really had no idea. And it was a source of actually great angst. And I'll tell you that this is not part of the talk, but really what happened was we were worried this was a terrible game and no one would want to play it. So we gave it to a bunch of biochemists. They weren't terrifically excited about it. Then we gave it back to them and we put a leaderboard on it. OK? They could all see each other's names and what score they got. We had to shut down the game in a week because it was like bringing all science to a halt. So I mean, yeah, it seems like a cheap trick, but that was really the moment when we thought, oh my God, we might be onto something here. Yeah.
So this may come later, so just tell me
Yeah.
if it's going to be. But now that you say this interface is being used by professionals, by
Right.
experts, do you have a sense of the differences of what they do with it and what the game players do with it, and how successful those are for different things?
Oh, that's very interesting. I don't at all. Yeah, I mean, I think that it's astonishing as this may seem, especially once you have an interface like this. Before Foldit, the way that you would do protein manipulation was you would write a script that would manipulate a whole bunch of numbers. And then you'd load it into it's like your compile, rewrite loop. So then you'd load it into your viewer, which would show you what you'd done. And that was basically how you did it. The latest thing was you could sort of pull something maybe. And here we have a full suite of interactive tools. Really, the idea was to make proteins toy-like, which is actually a technical term from game design if you know it, which means it means that its rules become manifest simply by playing it.
And can you give an intuition as to why people doing this interactively is better than a machine cycling through all the possible permutations?
Yeah. I'm going to talk about that in actually just a moment. Yeah. Yeah, that's a very multi-part question, though. We're going to talk about it in stages throughout the whole talk. OK. So the second reason why do you have a question?
I don't know that much about protein folding, so it's not clear to me why somebody could fold it and see that it was correct versus a computer, like the constraints that you might need in a computer to check that.
OK. Well, the second reason to crowdsource science is that humans solve hard problems. And so let me give you an example of exactly what you're all talking about. So these are 3 proteins superposed. The blue one is the so-called native conformation. So this is known to us through X-ray crystallography. The problem is that X-ray crystallography is very expensive and difficult to do. So that's why we'd like to solve this problem computationally. OK. The red protein is the best computer solution. And the cyan protein is the best folded solution. So this is designed by one of our players. And the key here is that little thing sticking out on the bottom there, which is so-called hydrophobic residue. So proteins are surrounded by water, and certain parts of them don't like to be near water. And so there's essentially a force which pulls them inwards. That is a hydrophobic residue. And so this is just the kind of thing that a scientist might look at this protein, and look at the red protein and say, this looks misfolded, because that residue should be pointing inwards in some way. It turns out that for all their vaunted computational power, the computer algorithm that we're comparing to here, which is one of the best in the world, never sampled that particular conformation. And so of all the conformations that it sampled, and there are hundreds of degrees of freedom here, so this is an incredibly complex space. It's also very hilly, by the way, in terms of the energy landscape. It just never sampled that conformation. But it's something that a human can just sort of look at and say, OK, this should be pointing inwards. And indeed, in the best folded solution, very similar to the true native conformation that was done. And now, you may be saying to yourself, OK, this can't be that hard, right? I'm going to write a little detector for the hydrophobic residues, and I'm going to pull it inwards, and so on and so forth. OK, if you can do that, I've got a little gift for you. It's called a PhD. I know you guys already have one, but these kinds of problems, why not have more PhDs, right? These kinds of problems are really the sorts of things that graduate students spend years on trying to solve. And so it's actually a very, very difficult thing to do to get these proteins to fold properly. And we published a Nature paper now, I guess, 18 months ago, showing that players were repeatedly able to do this better than sort of gold standard algorithms at protein folding. So that's very exciting. Yeah?
Follow-up to this, can you take a different approach if I wanted to make the computer better, which is instead analyze what are the perceptual features that people are keying off on, and then maybe in some sense learn from that in a way that might improve the algorithm design?
OK, you have a job. Yeah,
okay.
That's exactly.
You have a PhD.
Yeah, you do. Wow, you got everything. OK, I've got nothing I can offer you. Actually, that's what we're going to talk about next. So that's a very, very cool idea. So I'm going to give you another example from Eterna. This is our more recent game. This is a target shape. So remember that Eterna is the inverse of Foldit. So this is a target shape. We said we want you to build RNAs that fold into this shape. And here are a bunch of player designs. We call it a 5-bulged star, by the way. Here are a bunch of player designs. And as you can see, they look more or less like the star. They're not all completely perfect, but they're pretty darn good. I'll also note that these are empirical results, unlike Foldit. These are actual folded molecules. These are the designs from one of the top computer programs in the field. And it's not doing as well. And there's a reason for this, actually, which is that, forget protein folding, when we get into empirical science, it turns out that all of these beautiful energy models really have serious gaps in them with respect to what actually happens in reality. And so it's not really the computer's fault. The algorithms are actually fine. It's that there are mistakes in the energetic model. And these are mistakes that the players have been able to work their way around, basically through trial and error. So reason number 3 to crowdsource science. This is one of my favorites. It's a total rush. So I don't know, you guys, many of you are in HCI, so maybe you have more experience building things for people, but my background was much more in math and like algorithms and stuff like that. And the experience of building something which lots of people use is really unbelievably exciting. And I remember that when Foldit came out, we were on chat constantly and talking to the players the whole time. And one of the undergrads who was working on the project came in the next morning and excitedly told me about this exchange, which is that one of the players said, wow, you sure know a lot about Foldit, because he was telling him about the keyboard shortcuts or something like that. And the undergrad said, thank you, I actually was one of the programmers. And the player said, really? And the engineer said, yes. And then the player said, you are a god. How cool is that? When does that happen to us, right? Especially undergrads. So it's a very, very exciting thing. OK. Reason number 4 to crowdsource learning, to crowdsource science, is human learning. So as you know, machine learning is this giant juggernaut in computer science now. But I think that human learning is really a remarkable thing, and we should pat ourselves on the back. And so I want to give you an example of that here. So these are our earliest results from Eterna. And the way to interpret this is literally the first round. We had just released the game, and players designed RNAs for the first time, having no idea what they were doing. And so these red dots are what the players did. And that's one of their designs. You can see it's not terrifically good. Actually, the y-axis is better. Going up the y-axis is better. And then the blue designs are what the computers did. And so at this point, both the humans and the computers were working with the same incomplete energetic models and made many of the same mistakes. But the players had a trick up their sleeves, which is this cycle of experiments and hypothesis and results. And through this cycle, over the course of 6 months, the players got better and better and better at RNA nanoengineering. And these are not experts. It's pretty remarkable. And in fact, by 6 months into the project, the best computer design was worse than the worst player design on the exact same puzzle.
So they are experts.
Yeah, I guess, yeah, we can't say that anymore. Well, they're soon going to be co-authors. So I guess they are. So there we are at the very end, 6 months into the project. And so I was talking to one of the players about this. This is one of our top players in the world, Eli Fisker. And I said, I'm giving this talk, and I'm talking about man versus machine. Can you give me a little insight as a player what that feels like? And he said this sort of funny thing, which is that he feels sorry for the computers. When they started, they made all these mistakes that the humans rapidly figured out were dumb things to do when you engineer RNAs. And he actually goes into to explain one of the mistakes that the machines make. The tendency to put blue in the multi-loops, so on and so forth. Blue is C, cytosine. And so this is actually what started to get to your question. This is what started to get us think, wow, there's some knowledge that's been created here. And it would be very interesting to try and figure out what that knowledge is. And so basically, how is this happening? And that takes me to the 5th reason to crowdsource science, which is that humans create knowledge. And we just do this naturally. It's actually fundamentally part of being human, I think, even when you don't think about it. And therefore, it should be fundamentally part of crowdsourcing as well. So here's the timeline that I showed you earlier. These are the Eterna players getting better and better over the first 6 months. And right here in the beginning, when we were in our first couple of experiments, the players weren't doing very well. The computers weren't doing that well either. And this player called Dimension 9 made this blog post. And he said, I have been painstakingly going over most, if not all, of the new lab designs submitted by brand new players. And I was chagrined to find a dozen Christmas trees were submitted. Okay? So this is a Christmas tree, in case you didn't know. It's a sort of term that was invented by Eterna players. By the way, one of the fascinating things about these projects is that the players come up with their own technical jargon, very much like scientists do. This is called a Christmas tree, and it's a design which is made with only reds and greens, which are G's and C's. And we told them in the tutorials that G's and C's are the strongest bond, and indeed they are. So the players said, OK, well, I have no idea how to engineer an RNA. I'm going to use tons of G's and C's, which is not something that you ever see in nature. Okay? It turns out that for reasons that are a little mysterious, this doesn't work at all. And an RNA created with just G's and C's is not going to fold properly. It's a terrible RNA, even though the energetic model can't distinguish that. And so he said, Dimension 9 said, let us not waste even one of our precious design slots on these designs. And this was really sort of a clarion call to the community that was generated by the community itself, which sort of unleashed this torrent of a really deep analysis of what the results mean. So I'm just going to show you some examples. This is a blog post by a player called Chris Cunningham, who I learned recently was a grad student in computer science somewhere. Someone knew him. But anyway, and he is, so if you're wondering who our players are, and so he's sort of really taking apart this one of the results, nucleotide by nucleotide. This is another example of basically a paper that was written by one of the players called a meta-analysis of one-cross-bulge results. And here I'm going to zoom in on something. So this is written by players for other players with the sort of joint purpose of collaboratively beating this game. I find this pretty astonishing. And the thing is that when you look at this, I don't even fully understand what they're talking about. I mean, right? That's one of the hallmarks of science, that you're not entirely sure what they're talking about. And I'm sure it's meaningful. It's full of technical jargon. And this is the players are sort of doing this naturally. So it's a very astonishing thing. And that takes me to the sixth reason to crowdsource science, which is that I think that we're really creating new scientists. And so you asked this question about how can we learn what the players are doing. If they've learned information, we'd like to know what it is. And so we heard the players having increasingly sophisticated discussions on the message boards and talking about these concepts, some of which were known in the RNA literature, which is interesting, and rediscovered, and others which were completely unknown. And so we created this very simple thing called a strategy market. And it works like this. You type what you think is a positive or negative feature of an RNA into a little box on the website. And then our undergrads will code up those features, every single one. That was our sandbox. That was our security model. So you can't execute code on our machines because there's an undergrad in the way. We coded up every single feature that the players described, and we then tested them against a database, our large and growing database of empirical RNA data, and found which of those, using the Lasso algorithm, were most predictive, or what combination of them were most predictive. And we've actually just recently released what we're calling the Eternabot algorithm, and which is essentially a Monte Carlo RNA design algorithm which scores itself by the features which Eterna players came up with, some of which were unknown. And unfortunately, I don't have the slides on this now because this is so recent, but I can show you some results which indicate very strongly over the past couple months that Eternabot is outperforming all other algorithms, although not quite the players yet. Yes?
What features did the players have and what fraction do you use from Lasso? I mean, I'm just curious as to the signal-to-noise ratio in here.
Well, OK, so in total, I think this happened last summer. And over the course of the summer, we coded up 70 features, I think, between like 70 and 100 features. But it's a little bit more complicated than that because the features have sub-features in them. Sometimes they'll say, look at this and look at that. So we coded up 70 strategies, we call them. And then each one of them, essentially, what it does is look at an RNA and outputs a numerical score. And then what we did was Lasso found this optimal sparse combination of these predictors, which in order to predict the true empirical score that we get back. And then there's like 4 layers here. And then a Monte Carlo algorithm tries to design RNAs maximizing that score.
NASA step, did you feature select all of the 70 features that you got, or did you only use a subset of them?
We ultimately chose 5 features. Yeah.
That's great.
Yeah. And I'll tell you which one of them in a moment. It's pretty cool. In fact, right now. So this is one of the features that was discovered by the players. This is actually Eli Fisker again. And the fascinating thing about this was one of the features that was naturally chosen by the lasso algorithm. So it turns out it's very predictive of whether or not an RNA will fold properly. And it was completely unknown in the literature. And the feature is this. This is called a multi-loop junction right here. And here you see a C and a G. And what Eli said was, if you go around a multi-loop junction, your C's and your G's should be in order, either turning one way or turning the other way. But you shouldn't have them back to back. Completely unknown. No one knows why this works. But it turns out that it works very well. And this was a pattern that was essentially discovered by one of the players. So I think that this suggests a way of actually attacking these kinds of big data problems where there are gaps in our algorithmic knowledge, which is to convert these problems into games and just see what people do when they play them. This has been an idea that's in computer science for decades, but I think that really we're at this point where we can do this on a massive scale, and that makes a big difference.
Can you give us a quick intuition as to what it means for something to fold properly or not?
Yeah. So
how would Eli know, say he had flipped the C and the G,
right.
How would he know that that was crummy?
Well, so, OK. I don't know if this experiment was done specifically, but Eli could, for example, flip them and then submit that design for synthesis to see whether it worked or not. And the players do indeed design experiments to see whether their ideas are working.
I mean, I don't have a clear understanding of what it worked means.
OK, yeah, that's
It would be nice to get a quick overview of the game structure. You give it this, it gives you back that.
OK, yeah, sure. Well, if you want, so first of all, let's start with the question, what does it mean for something to fold properly? So this is an RNA. Each one of those little dots is a nucleotide. And the way all these biological molecules work is that they're defined by a string. And they always come out in a strand. And then they fold in the cell into some shape, which determines its function. So in this case, we can start reading from this end. And this sequence of letters, these are actually locked, because we need to lock them in order to do the experimental readout. But these nucleotides are locked. And then these nucleotides, G, U, U, A, G, A, C, and so on and so forth, is what was submitted by the players. So at the sort of raw level, when you hit submit, and the information gets ferried over here to Stanford, you will see that sequence of letters. Okay. And it turns out that for any sequence of letters up to a certain number of nucleotides, we can pay a company and they'll synthesize it for us, which is also astonishing. OK. What it means to fold properly. So these things fold Watson-Crick pairs. So A's bind with U's, G's bind with C's. Occasionally, there's a non-canonical Watson-Crick pair, GU. And so this is the design that we want to get. So basically, this says like nucleotide, whatever, 18 should be in a Watson-Crick pair with nucleotide 74. And 19 should be with 73, and so on and so forth. This is called a stack. And so this sort of matrix of what should be paired with what is a successful fold.
So that you give the player as their puzzle that description.
This shape, right, which is really just a description of pairs of nucleotides that need to be bonded to one another. Yeah.
So could you also talk a little bit about the onboarding process? Like when a new player joins the game, what do you tell them? What do you expect them to know? And just how do they start playing? Like, what's the gameplay?
Yeah. Well, do you want me to bring up the first tutorial level? Maybe that'd be the easiest way of doing it. Yeah. So I'm going to have to. I want to see what's going on here. So arrangement, mirror displays. What are my options here? Guess network access?
I got a computer.
Oh, great.
Somebody who's already in work has to give you a password.
Okay, yeah.
So we'll come back to this.
Okay, we'll come
Hold on
back
to that for a minute while he does that.
Yeah. Okay, but hopefully this is sort of the this is called the inverse folding problem, and hopefully this is clear. So we have this network of things that we want bonded to one another, and you have to submit a sequence which we have an assay which tells us whether or not it folded properly. You have to submit a sequence, and we tell you how it folded, basically.
Do you have some way to triage them?
So so this is actually an interesting thing. When the game was launched, we could only synthesize 8 RNAs per week. So there was a voting process, basically, which is a very fraught proposition. It turns out that players still did remarkably well. Then we went up to 16. So the super Moore's law of biochemistry is really astonishing. We went up to 16 RNAs per week. Then we went to 32 RNAs per week. And now we recently got some funding, and we're going up to 30,000 RNAs per week.
So when you test the folding, that's not a simulation. You actually build them.
Yeah, this is in vitro.
And then how do you read out how they folded?
So first of all, I'm not an expert on the wet lab side here. I'm hoping that my Stanford co-PI will just walk in right now. But basically, it's something called chemical accessibility mapping. And so what it tells us is for each nucleotide, it gives us a probability that nucleotide is bonded. We actually don't know to which nucleotide, but it's like a number between 0 and 1. And as it turns out, if all of the nucleotides which should be bonded are, and those which aren't, with extremely high probability, the shape has folded properly. And if there are some mistakes in the readout, we can predict what shape it probably folded into. So it's essentially a 1D readout of the RNA. And we can use that, we can sort of bootstrap that into a visual representation of the RNA, which we give back to the players. Yeah, you good? Let's see, Chrome, okay. Are you hearing this, hackers? Okay. Okay, here we go. So here we are online, and let's pretend that I was a new player. So you wouldn't actually get into it this way, because you'd be led directly in as a new player. So welcome. So here we are, literally starting at the beginning of Watson-Crick base pairing. Our first goal is to turn everything to G. It was worth every hour spent on that animation. This is also why it's worthwhile having friends who are musicians. I was like, Ben, can you make sounds for my new game? OK, so you get the idea. And I guess to make the sort of lab more concrete, this is actually we're starting to make switches, which is very advanced. But RNA lab, past RNA labs.
And you programmed this with an army of undergraduates?
Yeah. And actually, I should say, and one amazing graduate student, Ji-Hyung Lee, who I'll acknowledge at the end, but who basically started working on this as a first-year grad student, and now he's a fourth year, and he started working on Foldit. But he is managing a team across 2 universities with like tons of people in it. It's totally awesome. OK, so here is, for example, this was a F&M binding branches puzzle that we gave out. And the winner was, so this is the target right here. And the winner was Starry Jess, one of our players. So we can read about her right there. And it's called Aldo's Inspiration for some reason. And so here is the sequence that she submitted. So if we switch the view here. OK, so basically that string of G's, A's, and C's, and U's is what she submitted. And, oops. this is the readout that we got back. So basically, yellow means that the experimental assay found that it was not binding to another RNA. And blue means that it was. And so, and then this is the predicted shape given the experimental data. And what it tells us is that this is probably a correct fold. So that's what the game feels like when you get into the lab. Any questions about that? Yeah. Well, so I'm actually, believe it or not, the bubbles are part of the talk. Also, I just noticed the bug in the chat. OK. OK. So basically, through looking at hundreds of results like those I just showed you by Starry Jeff, this player, Eli Fisker, was able to put forth basically a very simple scientific hypothesis, and it turned out to be extremely predictive. Hey, Riju. OK, so if you think of, by the way, this is Riju Das, who's the co-PI on Eterna, and in charge of all the experimental stuff. So if you think about crowdsourcing as the process of using computers to sort of distribute lots of small tasks to many people, in many respects what's happening here is a sort of inverse crowdsourcing, where we're using inputs from thousands of people to teach computers how to do a task properly. But that's not all. So this section is called Creating New Scientists. Earlier in the year, we decided to have a summit meeting with our players and basically allow them to talk to the scientists and ask any questions they wanted about how the process works and so on and so forth. And so here's a little excerpt from it was over chat, by the way. So we just said, if you want to talk to the scientists, get online at this time, at this day. And the players started saying, look, 8 RNAs per week, it's a little bit slow. We'd really like to do things a little faster. Is it possible you could tell us what your experimental protocol is and give us some idea of how much the chemicals would cost so we can do a little bit of experiments on our own in the background? And this idea, this totally blew us away. I mean, we never imagined that Eterna could sort of transfer into the real world in that particular way. And actually, I'm not sure if anything has come of this yet, but it's become this sort of legendary idea in Eterna of backyard biosynth. And I think the key point that I'm trying to make here is that the existence of these games in society has really altered the people who are playing them. And that's just a very exciting opportunity in crowdsourcing we shouldn't lose sight of. OK, number 7. This one came up sort of serendipitously, breaking the rules. So when you're not actually designing real RNAs to be synthesized and validated, we let players play challenges. So these are little so-called in silico puzzles where you're just playing against the computer trying to fold an RNA properly. And what we discovered to our horror is that these players were beating our entire set of challenges at this ferocious rate. And right when we were going to run out of challenges, our in-house puzzle creator, Snehal, left for a job in the financial services industry. And we had no way of getting new challenges. So we did what's always the right answer in crowdsourcing. You guys know what it is? Crowdsource it. So we asked the players to make puzzles for one another, player puzzles. And all sorts of interesting, fun things came out. I mean, this is the RNA alphabet by Clawlin. And so this was sort of a step to just keep the players, one step behind us and keep the machine running. But we realized actually that in retrospect, that this is in fact a scientific problem in its own right, because the players were finding puzzles that they themselves could solve, that the algorithms couldn't solve. And so we turned this into a subgame in Eterna, where you see here are 3 major servers or RNA folders, automated algorithms. And the challenge of this game is to build a puzzle that you can solve that these algorithms can't solve, and ideally that none of them can solve. And so I'll give you an example, a very concrete example of what that means. So this is this little zigzag pattern. It's so simple. There's only like 8 nucleotides in it. It was discovered by Joshua Weitzman. And it turns out that one of the leading programs in the field, Vienna RNA, can't solve this. Okay? So we have these players systematically probing these algorithms for errors, basically, which is a really cool problem. Now, you might also say, now look, this doesn't seem so hard. There is a solution to this little design. It's just a G and a C or something. I don't know. Why don't we just teach the algorithms that little sequence, and then we'll fix the problem? And to that, I'd say yes. That's exactly the point. It was unknown that these kinds of little holes existed in the algorithms, and we're systematically finding them. So, you know, one of the themes of this talk is that when people think of crowdsourcing, often they think of sort of simple image and pattern recognition tasks. But I really want to convince you all that it can mean non-experts solving problems at the limit of human knowledge. Okay. Reason number 8 to crowdsource science is that there are a lot of humans. In fact, in the first 6 months of running Eterna, we had 26 man-years of work done for us for free. Continuous man-years of work. So there are a lot of humans in this world, and they work for free, which is awesome. And so this gets to Scott's question, which is why are they doing this? And honestly, a big part of it is score. And it's that if you set up these kinds of competitions, you know, it's almost like this caveman thing where if you have rocks in your cave, I want more rocks in my cave, even if there's no reason to have rocks in our caves. And it sort of reminds me, Luis von Ahn, who's well known in crowdsourcing, another professor at Carnegie Mellon, once told me, Adrien, don't just give them points. Give them lots of points. And that we do. We give them lots of points. But the other reason I think why they play, and a little bit deeper, is that there's a lot of science in these games. And so this is something that we actually got wrong in initial designs of these games. We tried to sublimate the science. And our idea was, oh, won't it be cool? You're playing this game which looks like Bejeweled just for fun, but secretly you're solving scientific problems. You know, we tricked you, we punked you. But it turns out that, in fact, exactly the opposite turned out to be the case in playtesting. The more science we put in, the more people wanted to play. And so here is just a little, I just snipped this off of the website. But I want to actually thank the teams who worked on the designs of these games because, in fact, science is sort of deeply, deeply embedded in how the games are presented to people now. And it's actually a very difficult thing to do, as I'm sure you guys can appreciate. And in fact, someone said a question about the bubbles going crazy. The bubbles are an example of this. So the reason why there are bubbles in the background is to sort of remind the users that this is all happening in water, which it really is. So that's one of the very subtle cues that we give constantly to players about the real underlying science here.
In that case, the points that people are getting maybe aren't just points. They're points that have meaning. And I'm stealing that pitch from last week's speaker from Yahoo Research because he was very skeptical of leaderboards for the sake of leaderboards. And his pitch was that leaderboards and other competitive mechanisms only work when they connect to things that have deeper personal and cultural and social meaning.
I see. Yeah. That's very interesting. I've heard a similar argument for achievements over score. The idea it being this achievements are sort of the new score in games, and the idea being that they get connected to sort of an emotional time, a specific instance in time when you got them. Rather than a score is like a bank account, right? It's like sort of fungible. But yeah, that's very interesting.
So it doesn't say that you should never have leaderboards or that you should never have score, but that scores only matter when it connects to something that you personally or socially or culturally care about.
Yeah, yeah. Well, I mean, that's very interesting. And in fact, you know, Riju and I think we're not really game designers at all. And I think, and numerous times people have said it would be really interesting to sort of do a deep study of what is motivating the players and what's really going on here. And I think we just haven't had time to. But I really think that is totally fascinating. And I want to know what the answer is. Yeah.
What are the achievements that players can obtain?
That's interesting. So there's a lot of achievements that you can get, and we're adding more. We're actually putting a lot of effort onto that now. But an example of, in fact, a very meaningful achievement is that you can't even get into the lab until you have 10,000 points. And that serves a number of purposes, one of which is that it's extremely motivating to get 10,000 points. In fact, if you look at the score distribution, there's like this peak at 10,000 where the player's like, I got in the lab, and then apparently they stop playing, which is, we're like, that's the whole point. Okay, but the other thing is that, so 10,000 points is an achievement. Well, the other purpose that it serves is that it sort of makes sure that we have an elite community inside the lab, which I think is actually very important for preservation of the culture of Eterna. It's a very delicate thing. It's actually very expensive, as Riju will tell you, to run experiments on behalf of total strangers. So you want to have good people doing the experiments. Yeah.
So it seems like with Foldit, a lot of it focused on answering these questions. And I'm curious what you think about science as not just answering questions, but asking good questions. Does the ability to create puzzles lead to creating good scientists in the way that you're teaching them how to ask good scientific questions?
Okay, wow, that is I love that question. So indeed, as we've in some sense handed more and more of the game over to players, we've given them more and more of the responsibilities of being a scientist, so to speak. And in fact, Riju, you even did something where you asked the players to edit a list of papers. Is that secret? Or, okay. I don't know if you were just being lazy or something. No, so indeed, we are sort of bringing the players, and especially the most elite players, deeper and deeper into sort of the inner circle of science. And one of our dreams is that why do we write the papers and they solve the puzzles? Let them write the papers.
You get to the top level, that's Stanford PhD admission.
Right. Or maybe Stanford PhD.
Without the admission.
Does the score then represent in some way how much knowledge they have or how good they are?
I mean, it's a score like any other, but it's correlated. I mean, you get it by solving puzzles. So, at the top level, the players are aware of a much more sort of nuanced. There's a much more nuanced sort of cultural landscape. So some players are acknowledged to be really good at designing experiments. Others are acknowledged that they write very clear blog posts that the other players read. There's really sort of a shadow scientific community here, where players are writing blog posts for other players and analyzing the results and stuff. So it's very interesting. Yeah.
So what about the demographics of the players? Where are they coming from? What are their backgrounds?
We don't know. We haven't done it. They come from all over the world. One of there's a fellow from Wired who's writing an article about some of the players. One of our top players is a New York patent lawyer. Another one's a librarian from Denmark. So, but we haven't done any studies yet. That's part. I think we'd really like to do that actually and understand who's playing and why. Yeah.
So Manas stole my question, so I'm going to ask another one. So there's this idea of you have 100,000 or more users. What is the critical mass? Do you really need 100,000?
That's a really interesting question, and So the question is, do we need 100,000? Do we really need 100,000 players? In some sense, much of the best work is being done by the most elite players. That's certainly true. So in that sense, we don't have 100,000 players of the quality of the top players. On the other hand, you could flip that around and say this is a wonderful thing. We've searched through 100,000 people and found a subset of those who are amazing at this very esoteric task. Whether there might be an inflection point, suppose we had 100 million players, would the results be 8 times better? I don't know. Or would they be the same? I don't know. So we just don't have enough data points in this space to answer that question yet.
You talked about your lasso method, choosing 5 features or 5 strategies out of 70. And one way to look at it is that all 70 were good and these 5 just were most predictive. The other, not so flattering, is to say that the 5 was a signal and the rest of it was noise.
Right.
Which interpretation do you think is closer to the truth?
I think. Well, I think that they're both. I think some of them were not predictive at all, and that's fine actually. But that's what science is. But some of them were, right? And I think you all know you have like 100 bad ideas for each good one, right? But some of them were, and they turned out to be predictive on blind tests, which were separate from the training set. And in fact, the players hadn't even seen the blind test. So we basically stopped the experiment and then ran a set of experiments on completely new RNAs using the features that the players had come up with. So indeed, yeah.
People to say why they chose that strategy? Was there any thinking of why does this work, or is it just the school hack that I figured out?
Well, this is like an audience of really deep thinkers. It's like, no answer I give is meta enough. I don't know. And I suspect that many of these sort of features have been boiled down from discussions on the message board, and that we could sort of trace their history if we were to do that. But really, you know, this work may seem like somewhat polished, but from the inside, what it feels like is just keeping the wheels on this car as all this crazy stuff happens. And so, you know, many of these things we would love to spend the time and see what are the players talking about on the message boards. Who are they? We just haven't had time. Yeah.
Yeah. Could you comment on / are you concerned about how the trend of your game is going towards kind of developing and reaffirming this elite status when that seems to almost contradict what you mentioned a few minutes ago about crowdsourcing, right, and how there's such value in just getting so many people, like you mentioned, 26 million users in the first few months. Right. You know, there's plenty of people. It seems that those are things that are kind of running contrary to each other. Is that something you guys have thought about, or is that a concern of yours? Or?
Right. You know, I think that there's different kinds of elitism, you might say. It is valuable to have people who are good doing certain tasks, and I am delighted that our top players are playing Eterna. I wouldn't really want to watch them play tennis at Wimbledon. That'd probably be pretty boring. I'm not bothered by the fact that there's a distribution amongst the players. That's, you know, we expected that actually, and in fact we explicitly went out to make these games to see if we could identify people who were very good at these tasks. But I actually think these are sort of anti-elitist, if you will, in the sense that the elites aren't the elites that you would normally think of. Our top players are not biochemistry grad students, and it's in fact very difficult to become a biochemistry grad student or a computer science grad student. Quite apart from having to be insanely intelligent, especially at a place like Stanford, you really need a lot of time, and you need to have had time to go to school, and these are just things that many people in the world don't have. But you know, the top players in Eterna are better than any graduate students who work on this project. So through a completely separate filtration process than that of getting into grad school, we've identified people who are very good at these tasks. And I think that's valuable. Interestingly enough also, well, I'll just play this video, actually. Yeah, this is the perfect segue. So it means a lot to players.
With a protein that is so misshapen, it's ugly and it's red, and to work through that to a beautifully streamlined structure where it's symmetrical and things are tucked in and nothing is hanging out of the sides. Yeah, that is sort of the goal, and that's what I like doing, to sort of help it in some ways to sort of be what it should be. I'm an administrative worker in the rehab team. I'm just answering telephones, working on bespoke computer programs, interacting with staff. It's very repetitive. When I go home, I become a different person. I just like to measure myself against something, and it's given me something that my everyday life hasn't given me. It's to just use abilities I didn't know I had.
Okay. So that was, at a time, the #1 Foldit player in the world, about a year ago. Those are the 10 reasons to crowdsource science, my 10 reasons for crowdsourcing science. And really what I'm trying to do in this talk is give you an idea that doing these kinds of projects is really not this sort of linear progression of simply creating a game and then having people solve your problem for you. It's really this very, very nonlinear thing, and all sorts of surprising things came out of it. Player puzzles, the features that the players came out with, backyard biosynth. And so I'm just very excited by this field and what's coming out of it. Here are a couple of, we're actually running out of time, amazingly. Here are a couple of questions that I think are interesting. This third question gets to, Leslie, yours, which is, can we get the players to write papers without us? I think it might be possible. And I think we're going to see major progress on this problem in the next 5 years. And if we can solve the protein and RNA nanoengineering problems, maybe we can save the world. So let me also thank some of the many principal people who worked on Foldit and Eterna. And in particular, since he's in the room, Riju Das, who is co-PI on Eterna, did all the experiments, lots of the game design, and of course, lots of the sort of high-level thinking. And then Ji Lee, who is the principal grad student on Eterna, and principal undergrad wrangler. And those are my 10 reasons for crowdsourcing science. Thank you.
How many of these proteins are there? Are you going to run, like, are there 1 million or 12 more? Or
Right. That's a great question. I'm glad that we used it. How many proteins are there in the PDB, you think?
In the PDB?
Yeah.
About 100,000.
About 100,000.
And how many have been solved?
Well, I guess all of those in the PDB have been solved. Actually, let me answer this a little bit differently. It's not a question of how many proteins there are, because one of the goals of nanoengineering is to design new proteins. And so it would be extremely slow to have to change one residue and then run X-ray crystallography or run various assays on it. So really the goal is to have computers give us instant feedback on what we're doing, on what we're designing. So in that sense, there are infinitely many proteins. And we'd like to be able to predict how any protein would fold. Right.
So in this talk, you gave us a good number of reasons of how you made things work for this particular task. What are some things you tried that were counterproductive during this design? Anything particular that stands out?
Sure, that's an interesting question. So one of the things that, I'll give you an example. One of the things that we thought players would be really good at was folding proteins with tons of residues, like 1,000 residues or something, and also folding it completely from scratch, which is called de novo. And you take this, this sequence, and you have to just completely knot it from scratch. As it turns out, and it's sort of like the chess or the Go argument, right? The more stuff there is, the greater the gap between computers and humans should be. That's what we thought. But it turns out that actually computers are much better than humans at de novo folding. And strangely, humans are better than computers when you're close to the right solution at actually getting to the right solution. And so that's something that it took us completely by surprise. And as we realized that, we started redesigning the games to make use of those human abilities, basically. So I think in many respects, the success of these games wasn't simply that we picked the right problem. It's that we literally just created them, and then we were fleet-footed in modifying them to sort of reflect what people were good at. Yeah?
Is hilliness required for humans to beat computers?
Well, I obviously, if this were a completely convex problem, if this were like solving a linear system, then we wouldn't expect humans to be better. I mean, humans to be better. I think it's also not the case, and this is something that people misunderstand sometimes, that you are just purely moving a little point in this insanely hilly landscape. That would be insane, too. So instead, Foldit, the tools themselves are actually search tools. You can drag things around yourself, but you can also push these buttons that launch a series of ferocious automated algorithms, which will test every possibility for this particular sidechain or run gradient-based optimization on the whole structure. So in some sense, the human's role is not to solve every number or even know every number. It's more like a general of an army marshaling these algorithmic troops to fold a protein. And that's really what it's like, yeah.
Other interesting problems in science or otherwise that you think could be crowdsourced but haven't been yet?
Yeah, that's there. We get calls all the time. So when we first launched this game, people were really skeptical, and they said stuff like, this is never going to work, and so and so. And now it's like the opposite. We get calls all the time from people who are in cosmology, and I can off the top of my head, I can't think, but we can talk about it afterwards if you want. From people who say, we have this really cool problem, would you want to work on it? And even within biochemistry, these are just two of many, many, many problems in terms of like drug discovery, structural drug discovery, sort of machine learning type drug discovery, so on and so forth. So yeah. Okay, probably, how much time we have?
We'll take
150.
10 minutes.
10 minutes, okay.
So I'm interested in the dynamics kind of amongst the players. From what you've shown, it seems like there's somewhat of like a supportive, collaborative community on message boards. Have you experienced them being like really protective of their work? Like any issues with that?
Yeah. Okay, so there's very much sort of community engineering in these games, and I don't know how we did it, but I would say that the most active players in the community are very respectful of one another and basically kind, and they're not, elephants in a china shop. At the lowest level, we've actually kicked out a few players. There's a little bit of like flotsam and jetsam in any community, so who are just messing with things. But we didn't do that much else. I guess the only other thing that we ourselves did was we tried to be very responsive to them. So if they asked questions on the message boards or if they needed help or something, especially when the game was launched, we really tried to be there a lot so that they had the sense of someone on the other end of the line. And it evolved into both these communities evolved into really like wonderful communities. And it's especially important because there's a lot of ways of hacking these games actually. And these are not foolproof in a game theory sense against various kinds of tricks you could play. Like voting is a perfect example. But we don't seem to have any voting fraud. Or if we do, who's ever perpetrating it is like a great scientist because we're getting like awesome results. Yeah, Leslie.
2 different questions. The first was with the success of things like Kickstarter. I was wondering if you say something about crowdfunding in science. Like, do you think that will have an impact on
crowdfunding in science? I can't say anything intelligent about it, but I think it's an interesting idea. And in fact, it's a great idea. And in fact, right now the model of Eterna is you have to solve some puzzles, but other than that, we will give you for free access to this experimental data, which is it's a difficult thing to get ordinarily. So it's really sort of a gift from us to the world in some sense. That's unsustainable at the level of like a big pharmaceutical company. So money is going to have to change hands. So an idea would be players could get more slots if they paid for them, for example. Don't worry, we're not going to do that, players. Actually, these things are very delicate too, because part of the community is this idea that it is free, that we're not going to try and take their designs and use them for drugs and stuff, which we've guaranteed to the players, and that we haven't accepted money from pharmaceutical companies, actually. So.
My second question ties into this notion of when you try to push forward crowdsourcing science, if you look at, for example, things which involve a lot of human subject stuff, then within the kind of, e-patients kind of human, quantified self-approach, there's the kind of notion of really taking on, more responsible to do this kind of science. And curious if you had any thoughts on it. Will crowdsourcing science go beyond just asking people on mechanical turf to do things to having people do stuff in their everyday lives to do science on that kind of scale?
Yeah, I definitely think so. And I think I don't feel like an expert on these questions, so I don't really know what to say. But I think Backyard Biosynth is an example of ways in which players have basically asked to start doing the science themselves and quite independent of us. So these games have actually sort of changed me in the sense of, first of all, seeing the outpouring of support for them, like the unbelievable energy, and most importantly, the skill of top players is far beyond anything I could have imagined. And I mean, it just changes my view of the world to think that you can throw something out there and you will find people who are this good at them. Well, I feel like I'm getting off on a tangent here. I want to tie this back to an answer to your question, but it'll come to me in a moment.
Do these then go into some Wikipedia-like database?
Yeah, so they go into a database which is not accessible through the game. But we're making it sort of more broadly accessible because it actually has scientific interest quite apart from the game. And the Eterna bot algorithm as well. It's just a new algorithm in this sort of an accepted space of within RNA biochemistry of algorithms that fold RNAs. And so we're making those accessible apart from the game. OK, I think we have time for one more question. I saw you first.
Do you have any idea who the top players are? Are they like closeted biosynth enthusiasts? Or are they just people who like to do puzzles or people with a lot of time?
I think they're not. They don't seem to be professional biochemists. Although we do occasionally get biochemistry grad students in the community. So I think that there are people, certainly you have to have a certain amount of patience to play these games, a certain amount of time. But there are also people who just really have a talent for this. I don't think in a million years I could have come up with the GC turning pattern, even if I'd stared at all the data forever. And so I totally understand the players in the sense that if you are very good at this and there's a community that's built around it which celebrates that, then you want to join that community and contribute in ways that you didn't really know were possible before. All right, thank you guys.
Thank you.
For more, please visit us at stanford.edu.